In this episode, I am joined by Robert Ahrends, who is Associate Professor at the University of Vienna in Austria. We discussed the main focus of his research, lipidomics, and the importance of standardization and quantification when measuring lipids. We also talked about strategies to analyze and interpret lipidomic datasets, and how lipids are not just energy storage or structural molecules, but also have more active roles such as signaling. Join us in this discussion to better understand how lipids are studied and how they contribute to our biology. The Metabolomist is the podcast where scientists connect to explore the unspoken issues in metabolomics research. I am your host, Alice Limonciel, and together we will examine how metabolomic data interpretation is done. I will discuss with other metabolomists how they do this work, how they plan, execute, but also communicate metabolomics. Welcome to this podcast.
Hello.
Robert, you studied biochemistry at the University of Gießen in Germany and got your PhD from the Humboldt University in Berlin. After that, you went on to work for a year at Agilent developing analytical methods, and then went on to Stanford for a postdoc in chemical and systems biology. And you returned to Germany in 2013 to be a group leader at the University of Dortmund. And since 2020, you moved to the University of Vienna in Austria, where you're now an associate professor at the Faculty of Chemistry, and your work combines mass spectrometry and data analysis to decipher the metabolome, especially in contexts such as cardiovascular disease and signal transduction. But you have other activities, like I know, for example, you have some community building activities in the world of lipidomics. What would you add to this bio?
So it was the Institute for Analytical Sciences, the ISAS in Dortmund. Not
yes.
the
Yes, sorry. Yeah, absolutely.
Yeah. In general, we are interested in lipids in a very holistic way. So we are interested in the lipid itself, but we are also interested in the metabolism around this lipid. So what enzymes are shaping actually membrane changes, signal transduction. And for us, it's very important to have always like a functional angle on this. So what does it help us if we're studying lipidomes if we do not find the root of the changes and put it in context what this means for a function such as a fat cell or a platelets, which are two main model organisms or entities we are investigating.
Mm-hmm. Yes, and this is something we will discuss today together. We will talk about one of your publications from 2018 about platelet activation and the role of the lipidome in that context. Before that, maybe more general notions about lipids. As we all know, if we work in biology, lipids have been poorly considered at the beginning of biochemistry. They're one of the last classes of molecules to be investigated with omics, for example. What do you think that is? Even though we have a lot of lipids in our membranes, but not only. Like, for example, lipids are a huge part of our brain, so you would expect that people would have paid attention a bit earlier. Do you think there's a specific reason why lipidomics took a bit longer?
No, I think there were great work done on lipids in the 80s and 90s. Mm-hmm. Yeah. However, their mass spectrometry was not key parameters of many molecular biology labs. And so with Britta Brügger and Andrei Shevchenko, Wolf-Dieter Lehmann, and who else was Kim Eckhouse, who were the pioneers of lipidomics and translated actually nanospray to the analysis of lipids. There was a very interesting publication around in sort of the 2000s from Britta Brügger, where she applied electrospray for a direct infusion experiment. And with this became more interesting for membrane biologists such as Kai Siemens and people like him, or Felix Wieland, which were also working in Heidelberg. They adapted then this for their own research on lipid transporters and things like this. And since then, it's becoming more and more clear that mass spectrometry is the tool to go for lipid analysis, because you can analyze a lot of things simultaneously, and you can put this in a quantitative context. And here's compared to metabolomics and proteomics, you have to do this quantification thing, because lipids are acting in bulk and not as single entities. For
Yes.
protein, it's very easy actually to put in context because you have a nuclear localization sequence on the end of the protein. So you know this protein acts in the nucleus. For lipids, you don't have something like this. And lipids are most often sticking in the membrane together. So if you analyze changes then, without standards, you do not know what the impact of this certain lipid is on the membrane fluidity, stiffness of the membrane, and other things. So reporting in concentration is key. And therefore, it took a little bit longer because at the beginning, not much standards were available. And now it's becoming easier because we have like big companies producing the standards and we're stepping forward.
And you're mentioning the importance of this, let's say, the composition of the lipids, for example, for the fluidity of the membrane. This is something also where we needed this level of detail in the analytics to be able to know how long the chain lengths were and how many unsaturations we had in certain complex lipids to have a way to interpret also the effects that it has. If you talk about membrane lipids, if you know that there's an increase in the number of unsaturation or a change in the whole family of complex lipids with a certain type of fatty acids that composes them, then you can start interpreting the effects it would have on the qualities of the membrane itself. Or
as deeper you can get, as better it is. There's a shorthand nomenclature which was developed by Gerd Liebisch and colleagues, where you go then from the lipid category level down to the isomer level of lipids. So at the moment, we are not at the isomer resolution. We are somewhere in between, where it's possible to do MS2 experiments easily and then get the fatty acids with a number of double bonds and chain lengths, right? And this brings us already big step forward, because with this information, you actually have an idea of what lipids are connected to the backbone. And this makes it easier to do interpretation on membrane physics, but also helps you to pin down if a certain lipids act as a precursor for signaling molecule. Of course, from there you can also go deeper down using ozone station or Pertero-Büchli actually to pinpoint the location of the double bond. But this at the moment, there are some instruments which started to deal with this. However, this is not broadly applied in all the lipidomics labs. There are some labs which are doing this.
At the moment, there's still a lot we can do with the method we have. So your paper that we will discuss is a good example of this, I think. So you really start from the global lipidome and then progressively focus on a specific species of lipid that is going to be the beginning of a whole pathway that we will discuss in a minute. But before we get into that detail, I wanted to ask you, what is a lipid for you?
As I started, there was the definition of a lipid as a hydrophobic biomolecule. But this is a problem now, right? Because we are dealing with all the water-soluble and water-insoluble entities at once, right? So if you're thinking about fatty acid, which has some hydroxy group, this will be definitely water-soluble. But if you go then to a cholesterol ester, this will be water insoluble. So you have to cross somehow, I think, 20 log orders or 25 log orders of magnitude in this water solubility. So I think people more and more say these are organic molecules to prevent to say they are hydrophobic entities. However, if you think in a classical way about this, I would still say this is hydrophobic biomolecules, because even if it's water soluble, it will be less water soluble than, for example, a phosphate or an ATP or something like this.
And this has consequences, of course, for sample preparation, doesn't it? If you prepare to do analytical chemistry to study lipidomics more than small molecules, for example, do you use different methods to prepare your samples?
Yeah, this is true. There are one-phasic extraction methods, but most of the people in the field are preferring like biphasic extraction protocols, like Blick and Dyer, MTBE from Matthias, which has the benefit to first precipitate all the proteins. But on top of this, compared to the monophasic extraction protocol, you can separate then more hydrophilic molecules from hydrophobic molecules. This doesn't mean that you have to throw the hydrophilic phase away. You can still analyze them, right? But you somehow separate out things were to put them in different workflows. Yeah, so we have proteomics workflows we can do there, the lipidomics workflows there, and then the metabolite workflow on the other side. This metabolite fraction or the more hydrophilic fraction still contains a lot of lipids, lysolipids, and other things will still be inside. They are highly phosphorylated, sugar lipids. Most likely you will find them, but you get a cleaner detection. You reduce just the background noise with this, and I would recommend to do this. Yeah.
From the background I have, I remember we had different processes for different omics. And I think the most important thing is to be aware of which method you use and where it might make you lose certain analytes. Because, for example, we tried to combine proteomics and metabolomics from the same samples, and we did that. But you have to be aware that when you use a special solvent or a certain method, then you might lose a certain class of metabolites or a certain part of the sample to the extraction method you have. And doesn't mean you can't use it for anything else, but you have to be aware of the limitations of your methodology.
Exactly. And there was a nice publication Christina Koman did from my lab in molecular cellular proteomics. And you have to take care of the partitioning in between the two phases. However, if you put a standard directly at the beginning inside lipid standard or metabolite standard, it's accounting for. You just have to detect the molecule and then it's accounted for. And this is a great deal actually doing quantitative analysis because you can calculate this then back.
You mentioned these membrane-forming lipids and also signal transduction related lipids. Do you want to give examples for people who are not too familiar of classes of lipids that would be more on the signal transduction side, or that would be less part of this structural world and more of the effective world that people might not know?
In general, if you think about how signaling can work, there are two different directions, right? So one is a signaling molecule binds to a receptor. And therefore it should in most cases be water soluble. So we are talking about oxylipins, glucan 1-phosphate and things like this. On the other hand, there's something, for example, a signaling pathway which is called the unfolded protein response, where the membrane is changed, and the change of the membrane, the distortion of the membrane is doing something with the dimerization of proteins. And then downstream is a signaling pathway triggered, which not directly involves the signaling lipid. However, it's still a signaling pathway. So if you focus on the first one, there all the different lysolipids. It doesn't matter where it's coming from, sphingolipid metabolism or from glucerophospholipid metabolism. There most often you find receptors, which lock as a transmembrane receptors, decoupled protein receptors. And they are doing signaling, various kinds.
And the lipids can be both the ligands to those receptors, and they can also be the effectors downstream of what happens once the receptor has been activated. For example, in terms of ligands, I'm working at the moment a lot with bile acids. And if you had asked someone 30 years ago what bile acids do, they would have just told you they just help us to absorb fat. And then in the late 90s, it was discovered that there were ligands for certain nuclear receptors, and that revolutionized the world of the research on bile acids, and suddenly they were interesting again. So we might discover other things like this for other lipids as well. We still discover relatively recently things that we thought we knew, and then find entirely new ways that they can regulate biology as well. Sorry, you were going to say something else.
So we maybe forgot something, right? So we have also phosphoinositides. They're activated by a pathway. They are chopped down, then becoming diacylglycerols or other lipid or metabolite entities, and then act again as signaling molecules on calcium signaling and things like this, right? So this is then somewhere in between membrane and binding. Lipid is binding to a receptor.
There are lots of possibilities. So before we go to the specific paper we'll discuss, I would like to ask you a generic question about how you handle data interpretation in general. Let's say you have a typical lipidomic data set. Do you have a strict workflow of how you analyze the data? Or do you tailor this to every paper and every study and question that you want to address?
This is a complicated question. We are tailoring this most often to the model system because somehow there's knowledge known. For example, if you're working in synaptic junction with the clefts, then you have to consider gangliosides and things like this. So if you are in the platelet regime, then you have to consider oxylipins and molecules like lysosphingolipids, which are known to do something on platelets. phospholipids, right? So it's a little bit tailored, and there we try the following things. So we have a direct infusion assay where we recover all the phospholipids, triacylglycerols, and cholesterol esters, so the bulk lipids, and then we have special targeted workflow for sphingolipids, for oxylipins, for PIPs.
These targeted workflows, they do special kind of combinations of lipids because they have specific structures, and combining them differently because of the way they are made, or what kind of specific workflows are you talking about then?
Yeah, the workflows are more or less analytical tailored, about accessibility. Extraction measures is the same. But for sphingolipids, you would like to get rid of the phospholipids, right? So you cleave all the ester bonds with an alkylic hydrolysis. So for PIPs, you would like to have it very sour, so you reduce the pH and things like this. So they're a little bit different requirements on the detection. So bulk lipids abundant, right? And then other lipids like the PIPs, very low abundant, and some certain sphingolipids also very low abundant. So there you go more the targeted direction, whereas if you go for general lipidomics into more untargeted discovery-like extraction by covering with standards certain lipid classes. You always being quantitative.
Do you often then go the route where you would start with untargeted to figure out which are the most relevant classes of lipids and then maybe go more towards targeted?
Somehow we're doing this. So for the bulk lipids, it's anyway untargeted. For the targeted things, you cannot really do untargeted because you're losing sensitivity. What we do, but we open the panel, right? So we're using Lipid Creators, the software tool which we are developed, and then we're putting a lot of information inside and do a screen. But it's not really untargeted. However, you can cover in some runs a lot of different lipids, right? This is modern speed of instruments. And then we shrink it down to a defined target list on the end of the day of lipids which are in the systems. But key is that we can do the permutations of the target list at the beginning to figure out what is in our sample.
This is to analyze the sample and then identify the lipids. And then let's say you have now a matrix of your data where you have all your samples or your lipids that you've identified. What do you do from there? Do you do more data-driven analysis where you do a lot of correlations between different sets of lipids? Or do you also work on a more knowledge base where you would use pathway analysis or chemistry-driven analysis where you group things per class? Do you have a typical workflow for that?
So we can create this matrix because we are quantitative, right? So we can get our workflows together. We're creating a big matrix, and then currently we do two things. So one is like more mathematically correlation-based driven approach using Pearson correlation, cosine clustering, gets the different clusters, and the different clusters using correlation and anti-correlation gives you then a network. So it's still a network, right? You can easily visualize what classes are there, what is changing there, and you can with one look say what's going on in the system. You don't rely and you don't lose information. So this is one approach, and the other one approach we going something which is we call the chemical space. So there's a model developed from Dominik Schwutke. Here, actually, you can calculate distances in between different lipidomes. And this helps also if you go across species which have different lipid entities, so different lipid species inside. So one are the species of animals, and the other one are the lipid species.
I know. I also feel like I have to explain which species I'm talking to very often.
But since you have this chemical space model, you can start comparing the things and visualize the things. Very important. At the beginning, we started, of course, with some reaction pathways and network biology driven things. However, we came very quickly to an end because with lipids, not all the informations are in reactome, are in kex. There are some things which are already now getting more and more established. However, there's still a lack of knowledge because nobody doing the hard groundwork in molecular biology to find out what substrate specificity a certain ceramide synthase has and what not. And this knowledge is missing, and this biology databases are not becoming better because the information is not generated. This is a problem. So therefore, we decided to go more data-driven approaches.
And this is something that you can afford as well when you have a relatively large data set where I think in the paper we're talking about very soon, it's like 400 lipids or something that were measured. So you have a matrix that's big enough that something comes out of it. Of course, when you measure 12 metabolites, unless they're very strongly related to each other because you chose them like this, you have less chances to find something interesting. But when you have a larger data set, then it starts to really be powerful to use this kind of correlation.
Yeah, but even if with this larger data set, right? So the 11 lipids, which are highly correlated to each other, will pop up in this data set as a very dense node. Yeah, and then you will see it straight.
Let's talk about the paper. The paper I wanted to discuss with you is entitled Identification of Key Lipids Critical for Platelet Activation by Comprehensive Analysis of the Platelet Lipidome. You're last and corresponding author on that paper with Oliver Borscht, and this was published in Blood in 2018. I wanted to talk about this paper because there are a couple of figures I really like about it. It's a good example of a common approach that is to take a large data set. And then I'm really mostly talking about the lipidomic data set. So there's a whole story behind it about the biology that I will let you tell in a second. The thing I really liked about the data approach in here is that you have a kind of overall description of the omic data sets with very tailored tools that allow us to look at it from different angles to have an idea of what are the important features in it, what is the spread of the data set in the chemical space and all these kind of things. And then another couple of figures where you compare with and without activation of the platelets to see what is the impact on the lipidome, and then look at one specific pathway and see by looking at the different species of the different lipids involved in the pathway, how you can actually follow the pathway when you activate the platelets. I found it really elegant. Could you say a word about the biological background about this study? Just what were you trying to look at? And why was lipidomics an interesting method to look at it in that system?
First, thanks that you liked the paper. Nice to hear. Why we did this? Because a lot of things happen in the platelet lipidome, right? Platelets are changing drastically the morphology upon activation. Fatty acid pathways, the PIP pathway, to the outside switching phosphatidylserines, different phospholipases involved in calcium signaling, linking this back to oxylipid, autocrine feedback loops to the entire system where lipids are responsible for. So a lot should go on, right? And the entire thrombosis field has a feeling that this is dynamically changing. Yeah. However, dynamically changing things means also instability, right? So you don't want to have something dynamically changing where you have to keep the things intact at least for a certain time, right? And this was like a little bit contradictory, and therefore we went in to do this atlas, a quantitative platelet atlas, and use different stimuli to actually look how big are the changes. What is the impact on the membrane? And there were some things which were quite interesting to find out. First of all, just % are changing within 5 minutes. So 5 minutes, if you have clotting, you poke yourself. How long are you bleeding? 5 minutes? Yeah, not longer than 5 minutes. Depends on the size of the wound. But you should not bleed as very long, right? So it must be a quick process. 5 minutes seems to be okay for us. And then we discovered just 20% are changing, and this is also stimulus specific. And there was also another thing actually very interesting that just, I think, I can't remember, 20 lipids making up 80 or 80% of the entire lipidome. And a lot of these lipids were actually containing lipids, and especially here arachidonic acid. And this was then the next step. And then we remembered all the teaching at the university, biochemistry, how is signaling of this oxylipins working. You need a precursor, and this is arachidonic acid. So we focused subsequently on arachidonic acid signaling. So thromboxane, prostaglandins, and things like this.
Yes, let's look first at description of the data set in general, this landscape view of what the data set is, what does it look like, and this in the paper is figure 3. So there are a couple of graphs, and the one you mentioned, for example, where you see that these 15 lipids are actually corresponding to about 70% of the lipidome in the platelets, and the rest is the other 380 something that you measured. So it's really intense. So cholesterol being the first component, and then as you mentioned, a lot of PUFAs. And you look at it, for example, you also compare human and mice platelet lipidome and check to see if you find the same kind of species in both. And sometimes it's the case, sometimes there are differences. This is also interesting when you think, okay, we might be interested in studying platelet activation in the mouse model. So we should be aware that there are things we will see there that we might not see in humans and vice versa. This is always the main thing you have to keep in mind when you use animal models to try and better understand human biology. And then there's this beautiful figure 3E where you break down the lipidome into different classes spread out and beautifully visualized with the different classes and the order of magnitude of concentration that you find for each member of those groups or subgroups of the lipidome. That really gives a flavor of the spread of concentration that you already mentioned before, and also of the variety of different types of lipids that we can look at. Because if, like me, you know about lipids, or like you knew about lipids before getting into this field, just from what you learned in school and on the benches of the university, this is not what I remembered from my classes, let's say 20 years ago, when you learned biochemistry and molecular biology. I didn't go into that detail. And I hope that now we're including this into the teaching. It's really a beautiful new world of biology that is super interesting to investigate. And yet in omic papers, you often have these one or two figures that give us the vision of how broad the data set is, where it can take us, and what makes the main components of it. So you also see the contribution of the different classes of lipids to the overall lipidome. And then the same thing is in the next figure when you compare then activated versus resting platelet lipidome, that you see then which are the classes that change, in which direction does it move. And one figure I really liked was figure 4C, where you make this very simple graph where you show the lipid-lipid correlation within class and between class, which is a very interesting thing to see, because you have correlation between one lipid and another lipid, of course. But if that lipid is from the same class, then that would make sense. Like you have a whole pathway or a whole metabolic biosynthetic pathway that moves in one direction. But when it's between classes, this can be for different reasons. So when you have a correlation between classes, it could be a typical example, I guess, is if they share a common fatty acid that they incorporate in different types of complex lipids, and then this kind of makes them move together because we have an excess or lack of a certain
exactly.
fatty
There's
acid.
something coming from the outside.
It's like one of the small building blocks is common to all those groups, and they all move in the same direction, which you can guess, or at least you can think of looking at it when you do this kind of simple correlation matrices, looking at what you can see within class and between class, for example. I thought this was a very simple graph to make, but it can be really informative. And this is something that's really specific to lipids. If you work in small molecules, you can make a similar kind of graph, but you wouldn't come to the same conclusions because they usually don't share the same building blocks.
Yep.
And then what I've also found interesting from a biological point of view is that when you activate the platelets with thrombin or with CRP, you don't actually get the same impact on the lipidome. So platelet activation is not just platelet activation.
It's true. There are different receptors, which a lot of them.
It's like it's platelet activation, but it's not exactly the same. And actually, in the last, or in the figure where you focus more on the oxylipins and the prostaglandins, then you see that it's only when you combine both that you actually get this really intensive impact on the arachidonic pathway. To go back to figure 4, where you compare activated versus non-activated, then you have, again, one of the most efficient ways of analyzing large data sets is this correlation matrices, and then finding clusters of lipids that correlate together, and then looking inside those clusters. And here I always focus a lot on the visualization, because I liked the way you visualize this. So you have the typical correlation matrix, and then you extract each cluster and open it to see what's inside on the class level in the next graph. And then you take these components of these clusters and put them in Cytoscape, right, to build the networks, and then see how the networks light up with the different activation strategies that you have. My main question, is it how you always do things, or do you do a lot of trial and errors and a lot of exploratory analysis and trying visualizations to find most efficient ways to show it in the best way in the figures? How does that work on your side?
This depends on the data set we are looking at. However, if we go through the data, there are certain key features which just jump into your eyes, right? But then it's a long way of proving this and connecting this to the greater lipid environment. And I think there, such tools as presented, like this Pearson correlation, cosine clustering, which are standard tools in omics, right? But together with these networks, it's quite powerful to visualize lipidomes. We don't do it always in such a way. So this is like just data driven. You could also, if they are very distinct, so say we would go, for example, to a deeper level, and the human plated lipidome and the mouse plated lipidome would be completely separated. You cannot do this anymore because these lipids are not shared. So you cannot compare A against B. So you must come up with something else up. So there, for example, we could use then the chemical space, where actually is the chemical feature of a lipid. Even if the chain lengths are different and somehow you're getting the same hydrophobicity, then this chemical space model would way better reflect what's going on in the system, because another class is taking over the function of the earlier described class doing something. However, if it's totally the same set, then this is a perfect workflow to go with. If they are more distinct, I would slightly change the direction. And yes, on the end of the day, of course, you have to adapt this in the best way to bring out what you want to say with your science.
Because once you have the result of the correlation and the components of the cluster, you could also make a heat map, for example, and you could show how the heat map lights up with the different activation methods. Would it be the same for you, or do you see a benefit of using the network? Because there is an inherent component of the network, of course, that shows the hotspots and that shows which ones are heavily linked to others.
If you would do the hierarchical clustering, you would consider everything, and then for certain components you would have a nice cluster on a certain point, as you can see it already in the Pearson correlation and cosine clustering on top. This is hierarchical clustering. It's not hierarchical, but it's a cosine. However, it's more or less similar, but then you have this cluster there, and then still you ask yourself what is going on there, how good are they correlated, and you don't have the cutoff. So with the correlation you can define a cutoff, just go inside and look for the highly correlated or anti-correlated things at once. You look at them at once. And with the other one you cannot look to the things at once. They are on different spots. And here you can do this at once, and on top of this you can put, by just mapping the classes onto this network before, you can also let the reader instantly understand what classes are involved. This you can do with extensive labeling, in your hierarchical clustering map, but I think this is already information overkill. So on one hand, we reduce information in order to get the right information to the reviewer at the right time.
It's a communication game in the end, because
exactly.
How long does it take to make an analysis like this? In terms of scale, what would you say in general? I think people underestimate how much time it takes.
The most challenging thing is to get the matrix done. If you have the matrix, right, then it's fast. Then you can talk about 2 weeks to get to something which you can present. And just to get some data out and how to look, this then some hours, because everything is scripted. In order that you are happy with this, I would say 2 weeks. But the data now is upfront, 2 months.
Of course. So I expected something in the range of months. Overall, after the experiment is done and everything, it does take a lot of time to process the data. Of course, it always depends on the question you want to answer in the first place. But still, even if you know what your question is, there are still different ways to get there, to get to the answer.
Sometimes it's straightforward and nothing is coming out, right? Most often we're using then more or less data, because it's sometimes you have too many perturbations and you are clouding something. And sometimes, we're switching to a chemical space model. And if we are really working on signaling pathway, so say we focusing on endocannabinoid signaling, which we did just recently, then we also have to go to the biology, go then also to pathway maps of this specific pathway, which is good mapped out, in a certain way, at least the enzymes which are involved. But then you have to do a lot of groundwork. And this then is very exhausting. You're easily going up to half a year just to understand what's going on with this hydrolases, which are
what kind of
regulated
groundwork?
different direction.
What kind of groundwork do you mean in that case?
Literature groundwork. So if
Literature,
you don't,
okay.
if you cannot explain it by mathematical model, cannot explain it by chemical space model, or cannot explain it with pathways, most likely the literature around this is around. You just nobody put it into a database, and then you just have to start reading.
This is long. This really takes a lot of time, and it's also I find it can also be difficult to do this in the long term and be able to include all the information together. Like you also have the risk of then getting lost in the literature because it's not the only thing you do as well. So you have to really be focused on the story to be able to keep those things together and organize them well.
Yeah, you have to also evaluate the literature if it's good.
Yeah, also.
Is this true?
Yeah. Is it true? Yeah, is now an interesting question. Is it true?
Yeah, exactly.
And how can I know that? Yeah. And then there's figure 5 in the paper where you really nicely show with a group of small bar graphs where you show the levels of PI 18024, which ends up being a key metabolite in this study, that you show is broken down into LPI 180 and arachidonic acid. And then arachidonic acid goes on to give all the prostaglandins that follow after that. And I liked this figure because it starts from a very simple reaction that is clearly seen to be happening when you activate the platelets. And then you have all the data in the nice little graph. So if someone who's really interested in the detail can go and check if this or that metabolite is increased or not. And then put in figure 5D, you put a drawing of a membrane with the different steps and where some of the lipids in the rest of the figure are in the membrane and how they lead to the next metabolite, which I thought is a really nice touch, kind of putting the story into one picture. So now we would just call it a graphical abstract, probably, like with a few modifications, but it's really nice to get an overview of what's actually happening in the cell, to not lose track of what is happening in the cell and of what actually we're talking about here. That is this molecule that is being broken down and what happens afterwards. I like that part very much because you find that in some papers, but it's not necessarily a common thing for people to add. So I thought it was a nice touch for comprehension also when you read the paper.
You come always back to this quantitative data. You can just do this with quantitative data,
of course,
right? Yeah, because then you can follow. So you have at the beginning this molecule count and ending up on the end with that molecular count. It's not fitting always perfectly, but usually you don't make big mistakes and do not go with assumptions because you know lot input, and then if you spread this out, the numbers must become smaller. This is a nice thing of quantitative biology on the end of the day. So this is systems biology, right? That you can follow the tracks. The other thing what would be potentially could do is like an isotope tracer experiment. Incorporate something isotope coded and then go down the tree, and always if you see you have this shift in the isotope pattern, then you know this fatty acid is coming from this precursor molecule. However, this is a shortcut to do this by comparing the absolute numbers.
This comes to one of the questions I wanted to ask you as well. I know you're a big defender of standardization in lipidomics. So what are for you the benefits of increasing standardization in lipidomics?
It's overwhelming currently. Yeah? There are so many people doing metabolomics or lipidomics, and the quality of the papers are very often not as high as they should be, if also going into key journals. Yeah, this is one thing. However, I also there's certain things which happens already in genomics, right? You can compare data sets, you can use public repositories to enrich your own data. You do not have to measure this again. This is standard in genomics, right? But it's not a standard in proteomics and in lipidomics, not at all. And that's why we are driving this. So our aim is, for example, is if we have then different lipidomics data sets in a quantitative fashion, that we can compare different species with each other, or a different disease type, and figuring out commonly changing things which are not directly driven by the disease, but rather that you have a systemic problem within the body. For example, certain ceramides which are changing in cancer, in cardiovascular diseases. And this is a systemic problem, what we have. But if we do not compare this different disease model with each other in a quantitative way, which is driven by standardization of data formats and databases and protocols for preparation of the samples, how to analyze the samples, how to use internal standards, then this will never happen. Yeah. And then we don't find biomarkers and we don't move forward with our knowledge of how the systems are working, then we are just stuck.
Yeah.
That's why standardization, I think it's pretty boring in certain ways. It's not like the exciting molecular biology, which is hypothesis driven. This is like very dry groundwork, but in order to step up to the next level of lipidomics, this must be done. Yeah.
I guess there's a whole community of lipidomics experts that are advocating for this.
Yeah.
And
that's why we founded also the International Lipidomics Society. There's the Lipidomics Standard Initiative. We also try to reach out to other consortia like Lipid Maps, MSCL, Neurofed Lipids, just to name some of them, and also to ICBL, which is more the functional driven consortium or people which are doing lipid biology, right? Or the Lipid League of the American Society for Molecular Biology. So these are all people which we try to get somehow together to drive this field forward.
And do you interact also on other topics? Because I guess maybe the standardization brings you together, but this really builds a big community now.
There are different interest groups. Yeah, there's this functional interest group, for example, right? Where people discussing lipid ontologies, like how this can work together, how we can enrich data sets. Yeah, it's technical driven. Yeah. There's no question, because it's always bringing it back to mass spectrometry. Also, the bioinformatics workflows is either databases, search engines, this reference materials is very technical. However, you need all of this to create something bigger and to understand the biology. If your analysis is based on the end of crappy samples producing bad data, then the assumptions are also not very good.
Yeah. In terms of ontologies, at the moment, there is not really pathway enrichment analysis available for lipidomics. But if that
there are some things. So there are, for example, Martin Molnar, who did line web. Yeah, this is going in the direction, and also I think Lipid Maps trying to increase this in their database, that they put ontology features inside. However, so far it's nothing like Go or something like this, right? Where you can go into this and getting nice things out. But this is also bringing us back to the bad annotation of things, right? The molecular biology experiment, which are not done to enrich this databases. Yeah, so it's like, sag mal im Deutschen, der Hund, der sich in den Schwanz beißt.
The dog that eats his own tail.
Yeah, I don't know if there's an English expression like this.
Could be similar. I don't know. Okay, then before we close, I would like to ask you, what is your favorite lipid?
Yeah, this lipid which is in this paper. Yeah, it's lysosphingomyelin, because it was totally neglected for a long time, and it's a great example how membrane lipids are somehow acting together with signaling molecules. Right, sphingomyelin is very important as membrane entity. However, if the system cannot cope anymore with the dysregulation, like here between ceramides and sphingomyelins, then they try to get rid of some lipids. And this is done via this pathway. The things are secreted, and then they're doing something on platelets, or you can monitor them in plasma, for example, as a biomarker, which later found out by another group. Yeah, I think this is a nice lipid. Yeah, totally.
And this works then in plasma? You can see this kind of changes as well?
Yeah, there are a paper on Niemann-Pick A, B, or C. I don't know. I cannot remember. But then there are monitors also in plasma. They could see this in the patient. However, the other symptoms are way more drastic than this lipid. So I do not know if you can use it as a biomarker. Yeah, and also these are most of the people which are affected are very young. So we're talking about babies and young children, and this is maybe not applicable to draw them in extra blood if it's obvious that they have already issues.
Okay, then thank you very much, Robert, for discussing with me, and all the best. And so,
thank you for your time. Have a great week. Yeah.
You too.
Thank you.
Bye-bye.
Yeah. Bye-bye.
Thank you for joining us in this discussion. I hope that this episode gave you new insights and ideas on how to plan, conduct, and communicate your own metabolomics projects. If you'd like to continue this journey with us, make sure to register for the Metabolomist email list on the podcast webpage. For regular news on metabolomics and data interpretation, you can connect with me, Alice Limonciel, on LinkedIn, where I post on metabolites, analysis strategies, data processing tools, and more. And make sure to check out our other podcast episodes on the Metabolomist website.